Good morning, everyone. My name is Debby Bogaert. I'm very pleased to be part of this session on microbiome and host immunity in early life. And the topic I'm going to talk about is born in a microbial cloud. And I have no conflicts of interest to report. So I've jotted down 3 learning objectives, and one of them is that we're going to talk about how the infant microbiome is shaped after birth and what is actually driving that. The second one is I'm going to show data on how the microbiome is interacting with the host, and then also on how this interaction seems to be related to respiratory health, which is a specific example I will take regarding this topic. And I just want to underline that because this is a virtual session, I'll try to keep it quite straightforward. I use data of ourselves only. Of course, there's many more high quality studies that can be found in literature, but for today, I keep it quite straightforward. So we are focusing, like many of you, on the first 1,000 days of life. And we have a particular reason for that. And that's, of course, because there's rapid growth and development in children in those first 1,000 days of life. And we know that if this rapid growth and development doesn't go quite well, that might have detrimental effects on health, both in childhood as well as long term. So that, of course, counts as well for microbiome development. We now regard the human microbiome as one of the organ systems in our body, and also that has been shown now to rapidly grow and develop in this very first phase. So that's where our studies are focusing on. And why is that so important? Well, we know, of course, most data have been derived from gut microbiome development. And we know by now, by research over the last 15 years, that the gut microbiome is quite complex and elicits many functions, amongst others, immune system development, as well as regulation. It's very important for many metabolic functions throughout our body, as well as important for proper development of barriers, including the intestinal barrier. But there's many other functions that the microbiome elicits as well. And basically every niche in our body and the microbes living in there have their specific function. And that counts from the tip of our nose through our stomach towards our skin and sexual organs. And very specific roles are elicited there by the microbiome. The interesting thing, however, is that this particular microbiome organ system is not developed yet at the moment a child is born. Basically, the majority of it needs to occur after birth has happened, and it needs to go quite fast and quite concise. But the interesting thing as well is that as the moment a child is born in a microbial world, that are the bacteria that it can use to assemble its own microbiome and to develop it further over the course of its life. And it matters, of course, where you're born, in a hospital or at home, how you're born, mode of delivery, whether you're put on the breast, whether you are being cuddled by siblings and grandparents, whether you go downstream to daycare, what season you're born, medication you're using, and so on, how and to what microbes you are exposed to, and therefore from what microbes this child can assemble its own microbiome. And that's why in theory, and the current data are really supporting that, all these factors matter to whether you can elicit a quite balanced and stable and resistant microbiome versus maybe one that doesn't have these properties as much. So we thought around 2014 that we wanted to understand better how that process happened and what was driving that and what the consequences were. So we started this Microbiome Utrecht infant study, which is a dedicated microbiome birth cohort where we enrolled about 120 children in pregnancy, allowing us to sample them directly after birth, as well as at day 1, week 1, week 2, and so on throughout the first year of life, and basically even up to the age of 6. And we obtained materials from multiple different sites in the body, from the skin, the nasopharynx, the oropharynx, and fecal material, to try to understand also these microbial developments in these different niches. And we did that at healthy moments, but also at times that these children had respiratory infections in the first year of life. To give us an idea, one of our primary outcomes was respiratory health, how these two were related. So this is the first data I can show you, which looks like a really nice piece of art that you can put on your wall. But basically what it shows is that every single dot represents the microbial community of one single sample. And to make that a bit more visual, what you see is that all these dots on this side in the brownish colors are showing the development of the fecal microbiota from day 0 all the way up to month 6. And in the blue one, you see the microbial development for the oral cavity, and in green, you see the microbial development for the nasopharynx. So you see that they start at the central point, which basically means they're seeded by the same microbes from the outside world, and then they take each their own route, adapting to the niche that they have arrived in. So then we decided to see whether we found correlations with the environmental drivers. And for the oral community, we found quite little correlations. We found mostly season, birth mode, and feeding type to be associated with how the microbial community developed over there. We found more for the nasopharyngeal community, where the crowding factors were most important, having siblings, going to daycare, followed by season, mode of feeding, mode of delivery, and antibiotic use. And for the fecal material, found sort of a similar set in slightly different order, and pets was an additional factor that seemed to be part of the driving factors of this community. So for today, I will zoom in on the respiratory tract because there were the least data yet available in literature. And I want to underline that the respiratory tract from the tip of the nose down to the lungs is quite a changing organ system. And it also means that the microbes colonizing this niche are changing from the nose through the throat towards the lungs. But what we can clearly define is microbial communities at each of these sites that are associated with health or with development of or actual presence of infections and inflammation. So different communities and also differences in biomass as well as low diversity is associated with more infectious pressure. So theoretically, how does that make sense? Well, you have a group of commensal microbes, and if they're more diverse, in theory, they're much better capable of competing for nutrients as well as through production of bactericidal metabolic byproducts with potential pathogens. Commensals also interact with the mucosa and thereby might stimulate them to produce antimicrobial peptides, secretory IgA, and so on. And the commensals are known, and all this information is mostly known from the gut, with innate and adaptive immunity at the other side of this barrier, which helps to develop adaptive immunity as well as inducing tolerance for the factors that you need to be tolerant for. But again, most of the information was derived from either animal work, bits and pieces, as well as from the gut. So we decided to also have a look in the respiratory tract, whether we could find evidence to support that. And I will focus within the respiratory tract on the nasopharynx because in another paper that we published recently, we showed that the nasopharynx in the age group under 1 is a quite good or a very good proxy for what's happening in the lungs at times of infection. So that will be the focus example for today. So back to our microbiome tract infant study, I will show you data on the first year of life. And what we try to do is look at the interaction between the respiratory microbiota, the host mucosa, and viruses that come and go in these children. So what we did was we characterized the microbiota for all samples available from basically day 0 all the way to month 12. And from these same samples, we had also generated viral data by these panels of qPCR that you all know from clinics, basically looking at adenoviruses, coronaviruses, RSV, influenza, and so on. From a subset of samples, what we also did was we looked at the gene expression profile of the mucosa, because you scrape off mucosal cells, you can look at their epithelial expression, and we used Affymetrix microarray for that. And that generates all these data, but also matched data, which are very valuable to really start to look on individual level at the interaction between microbes, viruses, and the host. So if you look at the correlation between these three data sets, you see something interesting. If you look on group level, what you saw was that there was the highest correlation between how your microbial community looked like over the first year of life versus the host gene expression. And there was less correlation for microbiota versus viral data or gene expression versus viral data. And if you look on the paired samples and the paired data, that was actually even more outspoken. There was this massive correlation between microbial community and host expression profiles, really suggesting these two are closely communicating with one another. These, by the way, are all unpublished data, so not ready for distribution yet. So let's first go back to the microbial community development data that we have already published in 2017. Here you see again the microbiota in these light colors in the early time points, day 0, day 1, and the developmental trajectory of the first year of life. And what you see is that you start off with oral microbes mostly, then you see all these gram-positives, Carina, Strep, Staphylococcus, different Carina, Dodicea gram and P gram, all popping in. And then at the second half of the first year of life, you see more gram-negatives like Moraxella, Neisseria, Haemophilus, merging into these communities. But these circles, which are basically representing the standard deviation of your microbial community at a given time, are quite wide, which suggests that this pace of development is different between children. They all go through the same routing, but the pace seems different. So we tried to look whether that pace seemed to be related to susceptibility of respiratory infections. So here you see the normal distribution of your infections over the first year of life with a group which have below average numbers of infections, 0 to 2, an average number 3 to 4, and an above average number of 5 to 7 infections. And if you then go back to their microbial developmental trajectory, the pace, you basically see microbiome age versus chronological age. And the reference group is the children with very little infections. So you see almost a straight line between microbiome age and chronological age. And then if you look at the yellow group, you see that at the beginning, sort of the first 60 days of life, they have a near similar age development. But then after the first infections, here represented by red, are occurring, you see that they have a slight acceleration of microbiome age. The interesting thing, though, is this group, the orange group, which have already in the first weeks of life an acceleration of microbiome age versus chronological age. So preceding their infections that they happen over the rest of the first year of life, suggesting there's a correlation between early microbial aging and a development of respiratory infections. So then if you look, what are the biomarker bacteria of this difference in microbial aging? So we get back to this early influx of these gram-positive bacteria, Dodicea granulum and different Carina bacteria and a bit of Staphylococcus, which is very early as 7 days in the 0 to 2 group. It's quite abundant and it's persistent. Whereas if you look at the 3 to 4 group, you see that that is less obvious a bit later and less abundant and less persistent, as well as in the 5 to 7 group. And at time of infection, here in this orange line at the back, you see that they're nearly present at time of infection, suggesting they're really biologically involved in this process. So what's interesting is that the abundance of these bacteria are actually inversely related to the abundance of potential pathogens that you all know quite well, Streptococcus pneumoniae, Untitled Haemophilus, Neisseria species, etc. Also suggesting that these microbes are containing these potential pathogens amidst them, preventing them from overgrowing and causing any inflammation. So let's quickly move to the viral data in this cohort. Again, you see here the data from day 0 to month 12, as well as the infection moments. And you see that actually already in the first month of life, quite a large number of children, I apologize for the small print, do have asymptomatic viral presence. And it's mostly human rhinovirus, and only later several other viruses kick in. And the first infections only occur from about this moment on. So definitely these guys are all asymptomatic preceding factual symptomatic infections. And if you compare them to the moments that children really have symptoms, you see also that that distribution of viruses is quite different, and especially less human rhinovirus, more RSV, as well as more human metapneumovirus here in green, for example. So we were interested in this early asymptomatic presence of these viruses. Do they seem to interact with the microbiota, and do they seem to be associated with downstream development of infections? So to have an idea about whether that happened, we wanted to see whether time to first asymptomatic viral presence was associated with your category of infections. And indeed, what we observed was that the children that ultimately have 5 to 7 infections have a much quicker time to first infection or to first viral presence compared to the other 2 groups. And if you repeat that analysis for human rhinoviruses only, which are the majority of viruses, if you've seen here, present in the early time points, you see exactly the same. The time to first human rhinovirus is shorter in the children that ultimately develop more infections. So that's an interesting feature again. So let's now quickly move over to the mucosal gene expression profiles. And what we did is something similar as what we've done with the microbial communities. We plotted the gene expression for a single sample in a dot in this 2-dimensional plot. And you see here crudely a couple of big clusters and some smaller ones. But the most interesting is to see here how that relates to the day and week of sampling. So the mucosal gene expression at day 0 is clustering over here, at day 1 over here, week 1 over here, week 2, month 1, and so on. So you see even a suggestion of a mucosal gene expression development over the first year of life in these children, like we've seen with microbial community development. So we wanted to see whether we could find these patterns of development in a bit more detail. And to study that, we performed a modular gene expression profiling. And what we observed was 4 big modules of genes that had a very similar expression profile over the first year of life. And module 1, you see over here, was low expressed at day 0, slightly upregulated at day 1, then downregulated for several months, and then only upregulated again from month 6 on. And these module contained genes involved in many different TLR cascades and neutrophil activation pathways, so basically in infection inflammation. Module 3 did something similar. It was downregulated for the first month and then slowly upregulated over the remainder of the first year of life, which basically contained a lot of genes which all were involved in interferon signaling pathways. And then module 2 and module 4 had sort of almost a counteracting expression profile. They were upregulated either from the beginning on or quite early and otherwise downregulated downstream. And these were 2 modules involved mostly in epithelial homeostasis, maturation, and differentiation. So then we thought, okay, is this expression of these modules actually also associated with your susceptibility to infections over the first year of life? So we did a similar analysis with the microbial community, and what we observed was that indeed the expression of these modules was associated with a number of infections, with an upregulation of both module 1 and module 3 in the children that ultimately got 5 to 7 infections, basically from very early on for module 1 and from day 0 on for module 3. So clearly, overexpression of these modules was preceding the first infections in these high susceptibility group. So since we've seen the same for early microbiome development and now also for early mucosal gene expression, we wondered whether these 2 were connected in line with what we saw at the beginning, that we saw a quite clear correlation between microbial communities and gene expression. So to investigate that, we performed a HALA analysis, which is quite complicated, but I only highlight the conclusions from this analysis. What we observed was that this overexpression of a group of genes involved in inflammasome activation and neutrophil activation were going hand in hand with high abundance of encapsulated Haemophilus species, and were downregulated at the moment that there was high abundance of Dalysigranum picarum and another bacteria that's seen a lot together, Yantinobacterium species. The second group of genes was overexpressed, and they were all involved in interferon pathways, again, with high abundances of unencapsulated Haemophilus species, and they were downregulated mostly with high abundance of Corynebacterium species. And there was a third group of genes that stood out. They were all involved in B-cell differentiation and other adaptive immune pathways. And they were going hand in hand with the upregulation of Moraxella species. And they were downregulated again with high abundances of Dalysigranum species. So I understand that's a lot of information, but if you try to link that all together, is what we observed was in this very early window, say the first 100 days of life, we saw that the microbial community development went hand in hand with the mucosal gene expression, and early gram-positive healthy commensal abundances were going hand in hand with low expression of inflammation and neutrophil activation pathways, which was then also going hand in hand with absence of early viral presence, especially human rhinoviral presence. So we were wondering whether the early presence of pathogenic species going hand in hand with more inflammation and going hand in hand with early viral presence and early initiation of interferon signaling was then sort of creating an environment where susceptibility for downstream infections was increased. It should be further investigated, but why is that so important? Well, we know that if children have a higher number of infections in early life and more wheezing, for example, that sets a tone for long-term health. So definitely worth to investigate. We will do that in our birth cohort. So we follow them up to the age of 6. We will check them for symptoms of wheezing, for lung function and airway reversibility, as well as for atopic constitution. And with us, obviously, many other research groups will work on that as well. So I hope to be able to report that further in the future. But if you go back to being born in a microbial cloud, I think the summary is that a child literally is born in a microbial cloud of microbes, and that it has to use that microbial cloud to assemble its own microbiome that then will be used for lots of functions, including immune maturation and metabolic functions, and they're at least very essential for their downstream health. So it's important to investigate that and see how that microbial community development in early life in general affects health and how we can use that for prevention and treatment strategies. So that leaves me to acknowledge the many people that have contributed to this study, my co-PIs of the microbiome birth cohort, Lieke Sanders and Marlies van Houten, my team that works in the Netherlands, as well as my team working in Scotland, and they both collaborate a lot and trying to decipher all these research questions that now emerge from the work we've done so far. Of course, I thank the funders, and with that, I want to close this presentation. Thank you.